So when we talk about research, there's two different routes that we can take with performing medical research. So there's what we call in vivo research, and then there is what we'll talk about in the next slide called in vitro research. With in vivo research, this is research that in medical testing that is done on a living organism. So if we were thinking about studying viruses, to do an in vivo study with a virus, you would have to study the virus in a living organism. So, I mean, obviously, if we're thinking about humans, the ideal way to study a virus in a human would be to inject the virus into the human and then see what happened. I mean, obviously, we're not able to go that route. So a lot of times when we want to study a virus, we either just have to do it, naturally in individuals that have already been infected throughout the with just exposure to the environment or transmission from other individuals. We can study it that way. But if we want to be intentional about it, we typically have to inject the virus into other living organisms and then make assumptions on how the effects that we're seeing in those other living organisms would translate to humans. So a lot of times they will use other animals, such as primates, swine, poultry. They will inject those animals with a potential virus and then observe the effect that that virus has on that organism, and then again draw assumptions based on how the virus would then impact the human population. They can also look at viruses in bird embryos, such as like chicken eggs, duck eggs. That's really nice because you can infect the embryo and then see what potential impact the virus has on embryonic development too. And bird embryos are sterile environments, so you don't have to worry about all the other potential contaminants that you may have to consider with live animal inoculations. But collectively, doing research in vivo is to do experiments inside an entire living organism. The two most common ways that viruses are studied in vitro would be through live animal inoculations and then the inoculation of bird embryos, two examples of living organisms. Me being an animal lover, I am definitely more of a fan of in vitro research, although I do obviously understand the need for in vivo research. But in vitro research is a way that we can experiment in a lab type setting. So it is typically done in a lab. Usually, if we're talking about viruses, it's done in the contents of a petri dish. So, I mean, ideally, if we want to study how a virus, again, infects humans, we would infect the human with the virus and see what happens. That's unethical. So what we can do is we can take human cells, grow them inside of a petri dish in the lab, and then we can infect those human cells within the petri dish and observe how the virus impacts the cells. What are the cytopathic effects? How is this virus changing the cells? And then obviously we can kind of make those connections to see what impact the virus would have on the human host as a whole. So studying something in vitro is to study something outside of a living organism in a lab setting, typically up within a petri dish. And this is what we're looking at here. These are little petri dish containers that have what we call cell lines in them, or cell cultures. And again, those would just basically be living human cell cultures. The specific cells that we're looking at here in this image are HeLa cells, originating from Henrietta Lacks. They are cervical cancer cells. The one benefit, I suppose, of cancer cells is that they do grow and divide rapidly, and they are relatively difficult to kill. So we can actually grow them in a lab very easily and very quickly, and then test to see how viruses affect them as human cells. So cancer cells, such as HeLa cells, are very beneficial and for studying viruses, but also all different other types of things, such as developing vaccines and studying toxin exposure to human cells, development of other different types of antiviral medications. So, but collectively, in vitro research is to study something outside of the human host in a lab. And our most common example of how we would utilize in vitro research is through cell cultures or cell lines, which is what we see here with the HeLa cells. I did post this very educational video where HeLa cells actually originated from. They did originate from a woman known as Henrietta Lacks, who passed away from cervical cancer. Her cells were incredibly unique in the sense that they would grow and divide extremely fast, and this was very beneficial to those in the medical industry who were trying to create cells that they could test for all different types of medical advancements. Unfortunately, those cells were taken from her body in the 1950s without her consent, and I think that this video gives us a really good insight into that and some of the awareness, and really puts her name out there, because she did have a very big contribution to not only the field of microbiology with the study of viruses, but really the field of medical science in general. So I really do encourage you to watch this video and take the time to learn about Henrietta Lacks. There's also a movie and a book, The Immortal Life of Henrietta Lacks, that I think are very good. I've seen the movie and watched the book, and they're very good. Right. So the next test that we'll talk about is a plaque assay, which is it's a type of in vitro experiment. It's done in the lab using a cell culture. But this is a way that we can see if a virus would be able to infect and destroy human cells. So what you do is you take a cell culture, possibly a cell culture of HeLa cells, and you would infect one of the cells with a virus. You would inject a virus into the Petri dish containing the cell culture lines, the HeLa cells. And then you would observe it over a 24 to 48 hour period. And typically before the virus is injected into the cell line, this would all be kind of colored in. So there wouldn't be any of this white. We would just see all purple from all the cells. However, if you infect a virus into the population of the cell line, and then you come back 24 to 48 hours later and you see these white areas that we're calling plaques, that's very bad. That means that that virus was able to infect the cell. Ultimately, it was able to replicate inside of that cell, produce more viruses that were released into additional cells, and then destroy those cells. So these big white areas that we're seeing, these plaques, if you will, these are areas where the cells have been destroyed, which is telling us that whatever virus we put in here, that virus is able to infect and destroy human cells. Now, if you infected the culture with a virus and you came back 24 to 48 hours and there were no white areas, everything was still nice and purple, that's good. That means whatever type of virus that is, it's not a virus that's able to infect and destroy human cells. But again, you come back and you look at it and you see these large white plaques where cellular destruction occurred. That's not ideal. That tells us, okay, this is a virus that is able to affect human cells. Obviously, would be able to cause human disease. The last thing we'll discuss are prions, which they're not viruses, so I know it may be odd that they're in this section, but they're also not cells, so I don't put them with prokaryotic or eukaryotic cells either. So they're kind of their own little thing, but we do put them in the virus lesson, but I do want you to make a mental note, they're not actually viruses. Prions are actually just proteins, but they are infectious and they can cause disease. So we all have prion proteins. We all have prion proteins in our brain, and when prion proteins are folded correctly, with proteins, they have to be folded in a very particular manner in order to be functional. But with prion proteins, when they're folded correctly, they are have a very important role of maintaining brain cell structure and support. So we all have prion proteins. They're likely folded correctly. They maintain brain cell structure and support. Prions only become problematic if they're misfolded. So what happens is when prion proteins become misfolded, they can no longer do their job. And what happens are the brain cells degenerate and die, and you eventually get these lesions in your brain where your brain begins to resemble a sponge and is no longer functional, and then ultimately can lead to death. So how would you get misfolded prion proteins in your body? The most common way is through ingestion of contaminated meat, specifically nervous system tissue. So if you ate from an infected animal like a cow, you could potentially get these misfolded prion proteins in your system, and over time it would lead to brain cell degeneration and death. This causes a disease that is known as Creutzfeldt-Jakob disease, or CJD. It's a type of what we call spongiform encephalopathy, which is a mouthful, but basically CJD causes brain degeneration. So you get these lesions where your brain is being destroyed and is beginning to resemble a sponge. That's why we call it spongiform. So here, this should all be nice and pink and colored in if it were a normal, healthy brain. But we can see these white areas called lesions, and this is where the misfolded prion proteins have caused the brain cells to degenerate and die off. Unfortunately, it takes a very long time, well, I guess fortunately, it takes a very long time for signs and symptoms to kick in. So once you would ingest a misfolded prion protein, it takes on average about 15 years for the signs and symptoms to kick in. So what would be happening during that 15-year period is your brain cells would slowly be degenerating. It just happens slowly over time, so eventually around 15 years, you would have lost enough brain cells where you would get to that tipping point where you would likely start to experience signs or symptoms. Now, while it takes a long time for the signs and symptoms to start, it has a very rapid progression, meaning once the signs and symptoms start, patients are gone within a year, and it's 100% fatal. There's absolutely nothing that can be done to treat it. It's just a protein. So you can't take antivirals, it's not a virus. You can't take antibiotics, it's not a bacteria. The only way to get rid of the infected tissue and the misfolded prions is to incinerate the tissue, and you can't do that to the brain and still have the person alive. So unfortunately, CJD is 100% fatal, and once the signs and symptoms begin, which are usually things like muscle spasms, delirium, hallucinations, memory loss. Once the signs and symptoms begin, all patients are gone within the year. So it is very, very deadly, and unfortunately it's very hard to get rid of this pathogen, and it has been transmitted to individuals through eating infected meat. It has also been transmitted through contaminated surgical instruments because it doesn't die or become inactive in response to standard sterilization practices, which typically would be to putting the equipment in the autoclave at 121 degrees Celsius for 15 minutes. That wouldn't get rid of the prions. You actually have to soak the surgical instruments in bleach for an hour and then sterilize at 121 degrees Celsius in the autoclave for another hour to get rid of the prions. So if you were just running standard sterilization procedures on equipment that was contaminated with misfolded prion proteins, you would not get rid of them, and you could potentially infect a patient. So have to be very cautious there. But we'll expand more on CJD later on this semester. Right now, I just want you to understand that it's caused by a misfolded prion protein that can lead to brain degeneration.